First of all, I should say that the discovery that I made needed to be interpreted in some way. If one makes an observation of even a minor observation, you want to explain it in the context of some aspect of biology, which I was then able to do. And having ruled out every other possibility that I could think of, in conversations with other people, it emerged that one of the things I hadn't ruled out was aging. And because at that time, the biology of aging was not really studied seriously by many people.
Certainly not on a cellular level, because Carrel had convinced everyone that cells do not intrinsically age. That was the whole point of his chicken experiments, right?
That's absolutely correct. What Carrel had allegedly shown is that if you put cells in culture, they replicate indefinitely, especially the cells he was working with, which are chicken cells.
Let me just interrupt you again. I also have here August Weismann's book. 1881, he writes this essay, The Duration of Life, and in it makes the prediction. He says that this statement leads us to seek the origin of death, not in the waste of single cells, but in the limitation of their powers of reproduction. And then he suggests that death takes place because a worn-out tissue cannot forever renew itself, and because the capacity for increase by means of cell division is not everlasting, but finite. 1881, I believe Weismann died just years after Carrel, a few years after Carrel had supposedly disproven the hypothesis. So your belief was, in those years at the Wistar Institute, 1960 or so, Weismann was forgotten. Carrel's views were dominant in science. So how in the world did you convince the scientific community that Carrel was wrong?
Well, you're correct. Carrel's views were dominant, not only because of the scientific work that he did with the allegedly immortal chick cell population. I should say parenthetically that the cells, unlike their description in the science magazine that you read, was not a beating heart. These were heart cells, but they were maintained in a plasma clot and weren't actually beating. Heart cells can beat for a while early on, but not after many years of replication and culture. The discovery of the paragraph that you read from August Weismann was not made known to the most of the scientific cell biology world, and certainly not to me until about 10 or 15 years after I made my observation. And I welcomed that. It was very prescient on his part to realize that, without any laboratory work. Of course, he was speculating. And then later to learn that Carrel was wrong. And the reason that's critical is that if Carrel was right, then aging would have nothing to do with what happens inside of cells. Aging would have to do with extracellular events, as I think I mentioned. But having torpedoed that idea, we were left with an explanation for Carrel's work, which was believed by so many people, and also because Carrel was one of the first scientists, at least in my view, to harness the power of the media. And he used the media to promote his ideas to an enormous extent. He even redesigned his laboratory to permit reporters to roam the balcony over his lab, which was further mystified to the reporters because Carrel believed that in order to reduce the contamination of cell cultures by microorganisms, which was a serious problem in the days before antibiotics, he had the entire work area painted black, and all of the workers there dressed from head to foot in black. So you had these black enclosed subjects roaming the floor of the laboratory looking like Ku Klux Klanners, only in black.
Or Al-Qaeda or ISIS or something.
Or some notorious group of people. And that lended the air to the field of cell culture, which until very recently had the air of a black art. And for many years it had that belief. And it was even driven further into that area of belief by what you mentioned earlier, and that is the realization that you could actually take cells from a human, put them into a culture vessel, culture them, look at them as they are living and dying and replicating, and to see a part of a human in a bottle evoked all kinds of unlikely beliefs in people who began to realize that.
Well, let me ask you this. So when you first wrote up your scientific paper, I assume you did not build a balcony and invite the press in to see your cells. So you submitted your scientific paper. How was it received? You go through peer review. What did the reviewers say?
Well, I won't answer that question until I tell you about two other events that preceded it that made the response to your question so important. Because Paul Moorhead and I, who joined me in this study, were very young scientists. We were in our late 20s, I think it was. We were told by, and I was told in particular by some major gray eminences in the field, that I probably made a mistake because what I had allegedly discovered torpedoed such a long 60-year-old belief in cell culture and in cell biology. So I decided to do an experiment that you won't find described in the paper. And that was to send cultures of one of my normal human cell strains to some of these gray eminences, in particular George Guy, who discovered the HeLa cell, and Harry Eagle, who was a very well-known cell culturist, and a couple of others, and told them that here we are in November, and I can tell you if you'll please accept these cultures, which they did do, that in 3 or 4 months' time, in January or February, these cells will stop dividing under your better conditions than mine.
And the significance of that is Eagle, of course, was, as you say, as an expert cell culturist, well established, I suppose, at that point. And George Guy had cultured the now infamous HeLa cells, which
Correct.
don't show an inherent finite capacity to divide. So that would be a very unusual phenomenon if he was culturing cells and they suddenly stopped.
Yes, I should say something that I failed to mention earlier, and that's a very important observation that I made simultaneously. And that is that normal cells have a limited capacity to replicate, and I also reported in that first paper and in this second paper, there were two, that cancer cells, like HeLa cells, were immortal. So it's important to understand that without knowing that normal cells are immortal, you would never appreciate the fact that it's only cancer cells that are immortal. Because prior to my work, it was believed that all cells were immortal. So that's a key concept that was part of the first paper. And also I should mention that in that first paper, we showed that the cells that I had grown were the most exquisitely sensitive cells to human virus replication. And I suggested that they be used for human virus vaccine production.
These are the
WI38, which was the one strain on which I focused all of my subsequent attention after the first and second paper, because I realized that all of these strains had essentially the same properties, and it's better to focus just on one rather than burden oneself on working with 3 or 4 at the same time. the suggestion that these cells be used for virus vaccine production in that first paper was very important because it was later found and to some extent known at the time that I published the first paper that there were serious problems with the cells that were then being used for the production of the first vaccine to be made on cell cultures, and that is the poliovirus vaccine.
Well, that's we want to get into the aging story as it's evolving, but for those who are watching that don't have a particular science background, so the concept, I guess, is that you grow human cells in a laboratory dish, and then viruses basically parasitize human cells. So this could be a way of growing, expanding virus outside of the human body. Effectively, and is that was at that time, I guess, your major goal? It wasn't to study cellular aging because you were just discovering it. Is that
yes, that's an interesting point because my interest in aging, although I suggested that specifically in that first paper, and unfortunately in recent years that has not become appreciated. Because of subsequent events that we'll be talking about shortly, the belief that the linkage between what I discovered and aging was made later by other people. That's simply not true.
Well,
my original paper states specifically.
How about this? Of course, this is 1968, I guess a few years later, but it couldn't be more clear. Human cells and aging. I think you're the only author on this paper.
Yes. Well,
certainly,
unfortunately, and I'll explain the reason why that has a shadow cast over it, or at least failure to recognize it in more recent years. I'll mention that in a few minutes. I suggested that because I had also discovered that in addition to the normal human cells, in particular WI38 and WI26, which preceded WI38, and were the main focus of my interest, that these cells are normal human cells with a finite capacity to replicate, and the finitude I suggested was telling us something about aging in my papers in the early 1960s, that these cells, because of their exquisite sensitivity to human viruses, and furthermore, because they were by every test known at the time and subsequently to be free of unwanted contaminating viruses, unlike the monkey kidney that was then used for the production of poliovirus vaccine and some other vaccines, that my cell populations had certainly had the capacity to produce vaccines under the most clean and uncontaminated conditions.
Well, that's, Len, oftentimes people in my field, of aging research focus just on this monumental discovery you made showing that Carrel was wrong, trying to overturn dogma, which is a dramatic and difficult sometimes things to do. But the fundamental importance of discovering that human cells, when cultured in the laboratory, age. But it seems to me that these same very cells, so let's say WI38, gave birth to two worlds, right? The field of cellular aging research, but maybe even more important as of today, vaccine production. I mean, as I understand it, WI38 have been used in more than one vaccine, and more than a few people have been benefited from that. Is that not true?
Yes, that was the most practical outcome of my work, and that is that, without going into all of the details, I further worked with vaccine manufacturers through the World Health Organization, and as of today, more than 2 billion people,
billion with a B,
people have benefited from vaccines produced in WI38, vaccines against rubella or German measles, polio, rabies, hepatitis A.
As the world
virtually
recognized
every
the value of what you've done, I mean, that's pretty remarkable to say 2 billion people and their health has benefited by work that you did in the laboratory back at the Wistar Institute.
That's correct, yes. Unlike the HeLa cell, which people have wrongly stated in print and otherwise, that has benefited more people than any other cell population, which is completely wrong.
Well, in what way?
HeLa cells have never been used for the production of a vaccine.
These are the immortal cells of Henrietta Lacks that
Yes.
we've heard so much about in books and media, but I mean, they've been used for research.
For research and for testing purposes.
But WI-38's been actually used and scaled up, and didn't I see that they were sent into outer space? And
that's correct.
I mean,
WI-38 has not only been used to benefit more than 2 billion people, it's also been used for research purposes wherever a normal human cell is required. It's also been used in virus detection laboratories where they look for viruses in the human population, and for many, many other research purposes.
if we get back to the aging story, which is so interesting in a minute, but another thing that comes to my mind is in recent years, we've had this debate in the United States and indeed around the world about the use of embryonic stem cells in fetal tissue research. Does the world recognize that WI-38 cells are the product of a human abortion?
There's a lot of evidence that many people do not. And lots of evidence that there are some quarters that want to keep it hidden in respect to the fact that the surgical abortion from a fetus in Sweden, I might add, WI-38 is derived from an embryo that was surgically aborted in Sweden legally, I emphasize legally, and that it has benefited more than 2 billion people. In the debates that have occurred since WI-38 was used for vaccine purposes in the 1960s, there have been several examples of very conservative individuals and conservative governments, like 2 or 3 of our governments in the United States, in which there was a prohibition to use any federal funds for research on human fetuses, aborted human fetuses.
But how could they, massive government funding must have been used, not only on WI-38, but for the production or the, for the distribution of these vaccines. Is that not true?
Yes, of course. It's absurd. I mean, the contradiction, the hypocrisy is world scale Hippocratic hypocrisy.
So, what, in a nutshell, on this, the use of fetal tissue or embryonic tissue weighed in the balance. People say, well, yeah, it involves abortion and there's this moral dilemma, but then you weigh in the balance the weight of human suffering, the weight of infectious disease. What's your advice to coming generations about how science and ethics and public policy ought to be made? What's the right path to follow?
Well, the right path is to trade the incineration of cells that otherwise could be used to benefit 2 billion people for that benefit, as opposed to simply incinerating them and not having safe vaccines to be used worldwide. That's one major example. I don't see any moral or ethical dilemma to using material that would otherwise be incinerated or in some rare cases buried when that material could be used to benefit living people. It's absurd to believe otherwise. And I'm willing to take whatever flack those who oppose this statement might invoke.
Well, my hope is that future generations will see the wisdom of some of the work that you've done in the past. Well, let's go back to aging. So you're using WI-38 cells initially to try to grow virus, clean cells to grow virus to to study the role of viruses, I guess in cancer as well as potentially for
Yes,
vaccines.
it should also be mentioned in the context of what impact this discovery had on different fields. It not only had an impact, a major impact in the field of human vaccines and in our understanding of human aging, but it also had a major impact in our understanding of cancer biology, because for the first time, it was appreciated that only cancer cells are immortal and not normal human cells, which gave rise to a whole area of research in cancer biology in which people tried to immortalize normal human cells by various technologies, in the belief that if we could understand how a cell becomes immortal, we will understand more about the biology of cancer. So that should not be overlooked either. Later on, I don't know whether you would like to move forward with this idea, but as it developed, one of my students, Dr. Woodring Wright, and I, although it was Woody's original idea in respect to the technology, we decided that after I had discovered that the cells had another property that was very important, namely the cells had a memory. If you froze the cells during these 50 population doublings, which you could do at each doubling, that is once or twice a week, at doubling number 27, 28, 29, etc., you could then go back to your freezer a year or two later and find that the cells remembered what doubling they were at the time of freezing and took off from that point to undergo the remaining number of doublings that would reach 50. Well, that was very critical. For the first time ever, there was evidence, and I had the evidence, that the cells had a memory, as I just described.
Well, let's talk about how that evolved into a molecular understanding of cell aging in a minute, but I'm still intrigued by this. You were bucking the trend. So you were saying that human cells age. Carrel had established firmly in the scientific literature that human cells, when cultured properly, if given proper nutrition and grown in the laboratory, would grow indefinitely. How did the scientific community respond to your paper? Did they welcome it with open arms, or did they welcome it by rebelling and saying you were wrong?
We wrote the paper in 1960, and I submitted it to a journal called the Journal of Experimental Medicine.
Which, by the way, wasn't that Carrel's journal, or it was the journal of the same institution, Rockefeller Institute?
That's exactly why I chose that journal to publish in.
Just put it right in front of their nose and say,
yes, of course.
Your institution and the Carrel legacy.
Yes, you published a paper that turned out to be wrong, although that wasn't my primary purpose.
So this is your Scientific American paper in 1968. This profile on Carrel is 1936. So this is about 28, 30 years later, right? Mm-hmm, mm-hmm. So you're saying the last 20 or 30 years worth of the legacy of Carrel was flat out wrong?
Yes, of course. And in fact, the paper that I sent to the Journal of Experimental Medicine was rejected on that basis. In a couple of 3-4 months, we got a letter from the Journal of Experimental Medicine from the Rockefeller Institute saying your paper is rejected.
Who was the letter from?
And it was rejected over the signature of Peyton Rouse, a colleague of Carrel.
Right.
And in addition to that, I can almost quote verbatim what part of the rejection of the paper was, and that paragraph read as follows. As anyone who does cell culture knows, that given the proper milieu in vitro, cells are immortal. Well, that's precisely what we had torpedoed for the reasons described earlier. And the paper was rejected over the name of of Peyton Rouse. So
basically what he was saying was, this has been repeated so many times, or is just so entrenched?
He didn't give any reason. It was clear that it was so entrenched.
Yeah.
He also said that the suggestion that this has something to do with aging is, quote, notably rash, end quote. And so we, of course, Paul and I were very disappointed. We resubmitted the paper to Experimental Cell Research, where it was published almost immediately without change. And I should say that it has been cited in the scientific literature almost 10,000 times, which is,
as I recall, it's one of the most
cited papers.
ever scientific papers in the history of science, which is congratulations on that. It's nice to be cited. I don't think my papers are cited quite that many times. I suspect that Carrel's papers aren't cited that many times.
Not anymore.
Not anymore. But maybe perhaps some of it too was the fact that he was so visible, as you say, publicly.
His image, his notoriety in the public press was enormous. There were New Yorker cartoons based on the escape from a test tube of the immortal chick heart cells that then replicated to cover the city of New York.
That giant, the blob that ate New York.
The blob that ate New York, and there are many science articles and movies made about this, science fiction movies made about this alleged phenomenon. One of the things that I recall vividly is a headline in the New York Mirror, I think it was called at the time. The paper is now defunct, but the headline said that if Dr. Carrel kept all of the cells that had been produced from his culture, now 40 years old, it could produce a hen so big that it could cross the Atlantic in 2 strides. Okay. And that was the headline in the newspaper.
So the sensationalism we see in the press today isn't all that modern of a phenomenon after all.
No.
That certainly was a sensationalistic thing. And there's a really interesting book called The Immortalists about Charles Lindbergh, also very much in the news in those days, joined up with Carrel, and as it says in the book, Lindbergh was convinced that together they could understand aging and potentially stop aging and death itself.
So that's another example of Carrel's technique, I guess you would call it, in obtaining as much public interest in what he was doing as possible, because after Lindbergh did his solo flight to France and came back a hero, Carrel offered him a job in his lab because he argued that that Lindbergh's knowledge of engineering would provide the information he needed to engineer the circulation in isolated human organs to keep them alive for long periods of time, presumably, and then use them to transplant in people who needed a healthy organ.
Sort of a very primitive, perhaps, version of what today we would call regenerative medicine.
Yes, to some extent that's true.
Before we move into the more modern era and how cellular aging took fire, because it really has become a major area of research around the world, what do you think happened in Carrel's lab? So, as I understand it, chicken cells never escape aging. I mean, the fact that he used chicken cells to grow cells and said that they would replicate without limit had to have been wrong. It couldn't have been a simple mistake. What's your best understanding of what happened back then? How could he get it so wrong?
Well, it turns out that we have now several sources of information that provide a good answer to that question. One of the answers came from one of Alexis Carrel's technicians, who I met under accidental circumstances once when I was giving a lecture at the University of Puerto Rico, and a woman appeared after the lecture to introduce herself as a former technician in Carell's lab, and explained what had happened to me at that time. And other people have also reinforced this probable explanation. And that is that the technique used for culture at that time necessitated the use of chicken embryo extract as the source of the nutrients for this so-called immortal chick heart cell population. The heart tissue at that time was not cultured the way it is today. The tissue was cultured as what we call an explant. That is, a tiny fragment of chick heart is embedded in a clot composed of chicken plasma and an equal proportion of chicken embryo extract. And the cells then migrate from the tissue peripherally, and when they migrate to the point that they reach the perimeter of the clot, they are then cut into 4 pieces, for example, with sterile scalpels, and each quarter put into a new culture vessel and cultured under the same conditions. And this was continued by Carell allegedly for about 40 some odd years. And what apparently happened was that the primitive method of preparing chick embryo extract daily at that time by Carell's technicians, which was to run the embryos prior to bone formation through the barrel of a syringe, and then that mashed up material would be put into a fluid and then centrifuged. Well, in those days, centrifuges were very primitive and tantamount to swinging the material around your head a few times in a bucket. So that under those circumstances, the cells, living cells in the extract, would not all fall to the bottom of the vessel, but many would remain at the top. And by removing the fluid daily to feed the culture, you would be introducing fresh cells into the culture on a daily or weekly basis.
So the chicken broth, or the chicken soup, so to speak, that they were feeding the chicken cells themselves, itself contained chicken cells, is what you're saying.
Correct, yeah.
So they kept adding new cells to them.
It was the chicken soup of gigantic proportions.
Yeah.
It not only cured everything, but it added fresh cells to its culture.
You had the privilege of meeting one of the technicians that was actually hands-on doing this. Did she imply that this was an artifact that no one was aware of? Or did she imply that perhaps Carell, since he was, promoting this idea and was getting notoriety, that maybe it was with his knowledge and consent?
I don't recall her specifically giving a reason for that. What was implied was that he was being protected by his technicians from knowing about this fact, because he had succeeded in attracting so much worldwide notoriety that to have this discovery made known would be an unbelievable embarrassment to him. That's one theory. There are others who have looked into this very seriously, who claim otherwise, who claim that he did know about it. But I don't know that we have any hard evidence either way to come to a conclusion.
So the lesson always seems to be that seeking truth is always the best path.
Sure.
And the contrary phenomenon, that when science doesn't follow your favorite paradigm, sometimes that's the most interesting discovery. And so you're finding that human cells aged turned out to be the most exciting story of all. And if he had observed that,
yes,
he would have been the father of modern cellular aging research, rather than...
That's correct. Well, there is another example of that, and I was faced with this problem as well. One of the leading cell cultures at the time that I made this discovery was a man by the name of Theodore Puck. He was at that time at the University of Colorado. And Ted Puck was a giant in the field of cell culture. Furthermore, He was working with normal human cells, although he didn't know that they were normal at the time that he did all of his work. His work preceded mine by a year or so and continued during my work. And he was working with human fibroblasts. And in fact, he published in the Journal of Experimental Medicine, which is another reason why I sent my paper there first. However, he believed that his success in growing human cells was because of the extraordinary attention that he paid to the culture media, and then specifically the serum that was necessary to be added to the chemically defined portion of the medium. And you will find in the Journal of Experimental Medicine, which his work is reported, several pages of 4-point type describing the extent to which he went in testing serum. I can tell you that I never tested any of the media. I bought it commercially and used it right off the shelf. But because of his stature in the field, before I had written this paper, fortuitously, I learned that a big meeting, the American, the American Society of Biology, had their major meeting in Atlantic City, New Jersey, which was 80 or 90 miles from Philadelphia. And he was to be a featured speaker at one of the main events. I decided to go to that meeting. He gave his presentation describing his work with human cells, and at the end, questions were posed, and I got enough courage up to raise my hand and ask the following question. Dr. Puck, have you ever observed that the cultured human fibroblasts you're working with ever stop dividing in culture? Oh yes, he responded immediately. That happens often. We simply go back to the freezer and reconstitute them, use them again. It must be because the medium is deficient. Well, that nailed it for me. In addition to which, and I failed to mention this earlier, the responses I got from my colleagues to whom I had sent cultures, leading figures in the field, responded in the month that I expected them to respond, saying that the cultures I sent to them had stopped dividing. So I figured if I was going to go down in flames after I published this work, I would have some very popular company. And that was one of the major reasons why I decided to publish a paper.
You still had some doubts at that point.
Because it was against the dogma, and I was being ridiculed by many people who said, Hayflick, you're wrong. You're going to ruin your reputation if you publish this paper. And that was a frightening thing to consider.
I can easily see that in several respects. One, you're biting this dogma out of a prestigious institute like the Rockefeller Institute. Second, you're talking about aging, and there's always been, at least in my career as a scientist, this expansive skepticism about studying aging.
Oh yes, you're absolutely correct. Sure.
And must have made you a little nervous as a
well, as I mentioned earlier, I entered the field kicking and screaming only because I was left with aging as the only tenable argument to explain what I had found. And at that time, I can say with confidence, there were no more than a half a dozen people in this field, in the field of aging in this country, and an equivalent number proportionally in other countries, especially in Europe, who were studying the field of aging. And most of the studies were descriptive. That is, what happens when humans or animals age. Mm-hmm. Very little was done, or could be done, in fact, in respect to the cause of aging. That is, the fundamental biological cause of age changes. That was almost totally ignored, although there was a belief that the fundamental cause had something to do with radiation or ultraviolet rays or stress or something extracellular.
And probably in part because of Carrel, don't you think?
A lot of it largely due to Carrel, yes.
He taught that cells taken outside of the body and with media changed and so on suddenly stopped aging, so, or at least they didn't age.
They didn't
And
age.
so wasn't then the idea that maybe some hormonal thing, endocrine thing?
Yes,
I'm
many
thinking
people.
Voronov and with his monkey
Monkey glands.
testicle grafts and so on, wasn't this part of this idea that maybe there's some hormonal deficiencies that cause the
Yes.
deterioration of aging? Not that it's intrinsic to cells.
Yeah, there's very little that you can think of that was not invoked somewhere along the line historically for as being the cause of age changes.
I guess I'm thinking if Weissman's idea that his prediction, his theory, that somatic cells had a clocking mechanism or an aging mechanism, and if Carrel had actually paid attention and noticed that his chick cells were also aging, maybe we would have seen a much earlier adoption to this idea that aging is an intrinsic phenomenon that involves cells as well, and maybe less of a protracted red herring of chasing after a lot of these other associated theories that never really went anywhere.
Yeah, that's a very tenable argument, and I would subscribe to that belief.